cloning can be a very interesting topic, or it can be the topic of an ethical debate because there's lots of questions surrounding whether it's right or not to be able to genetically alter an individual to create an individual that's genetically identical to it. But cloning, as a definition, is actually the creation of an organism that's the exact replica of another organism. But it's not like putting an organism into a photocopy machine. You actually have to start this organism from the embryonic stage, where it just starts as one cell, which starts to divide, and then it will divide and become clusters of cells, and then eventually form organs, and then it will become an individual. So it has to go through that whole growth and development stage. And there's two types of cloning. It's artificial embryo twinning or somatic nuclear transfer. And artificial embryo twinning is the same process that actually forms identical twins in the womb, but this is done in a lab. So an embryo is manually separated. So here we have a picture of an embryo. You can see the cells are dividing by mitosis. You have lots of cells, and what happens is these are going to be manually separated using tools in the lab. And then each cluster of those cells is going to continue to divide. And those are going to divide until it forms two individuals. So you end up with two individuals from this one cluster of cells. And these individuals are going to be genetically identical. Somatic cell nuclear transfer, or SCNT, was the process that was used to create Dolly, the sheep, in 1997. And to create Dolly, they actually used a somatic cell, which is a cell other than a sex cell or gamete, an egg or a sperm cell. Somatic cells can be cells from the skin, from your liver, from any cell that is not a sex cell in the body. And what they did was they took a donor cell from a sheep's udder and they took the nucleus out of that cell. Remember, the nucleus is where all the genetic information is. Then they took an egg cell from an adult female sheep, and notice this sheep has a black face. This one does not. They took that egg cell and enucleated it, or took the nucleus out. Then they combined the donor nucleus of the first sheep with that egg cell, the empty egg cell, with an electric shock. So they fused that, and then it started to divide. And that was a normal process. Now we have a cluster of cells called an embryo. They took that embryo and placed it back into the uterus of a foster sheep. Notice that this sheep has a black face. So they reimplanted this embryo into the sheep. This sheep carried the embryo to term and produced a little lamb. And this lamb was the clone lamb that was genetically identical to the donor sheep that the nucleus came from. And they named that little lamb Dolly. Let's talk about stem cells. And these are cells that are unspecialized, which means they don't have a job yet. So what happens is, well, we can actually add hormones to them so they take on specific jobs. Our cells start to divide if we go back to Dolly and this group of cells here in the embryo. These are actually all stem cells. They don't have a job yet. They'll start to specialize later and become differentiated. So what has been figured out in the lab is that these cells can actually be used if you take these cells and then add the hormones that are needed. You can form different organs and tissues from them. So if this can be done, then maybe it can be used to treat different medical conditions. And they talk about this as therapeutic cloning. So here we would take an egg cell and a somatic cell, and then we'll take the nucleus out of the egg cell and take the nucleus from the body cell and insert it into the egg cell, get it to divide, and then introduce that to form an embryo. All right, now we have all these cells. So now these cells, we can introduce hormones to them and get them to take on the jobs that they need to take on to treat medical conditions. So the question is to clone or not to clone. So there's some pros and cons about it. Good things with cloning would be maybe cure infertility. People that can't have children, or cure patients with life-threatening diseases. Through therapeutic cloning, maybe grow spare body parts, and possibly protect endangered species. Things that are kind of cons for cloning, it's unethical. We're kind of taking over the natural role that reproduction has by being able to produce these organisms that are genetically identical. And, maybe this technology could get into the wrong hands. The debate often comes up, that science is meddling with, nature and that it's not right. And even though a clone is produced and it's genetically identical to an individual, it does not influence the clone's behavior. The environment that that clone is in is what's going to influence the clone's behavior. And the process is also very expensive.